Good evening. We would like to start, and today we have Chaya Kalcheim, who is a professor at the Department of Medical Neurobiology, and also this is at the IMRIC, at the
Institute for Medical
Research, Israel, Canada, and a member of our new brain center, ELSC, who is working at the Faculty of Medicine since I remember myself, I think.
A little bit later,
because she's younger, and I'm very happy to have you here. And she will talk about, Chaya is a little bit concerned about how much we know about development. So I asked her to give more background. For those of you who are experts, please don't expect today the deep science, but more...
Why not?
No.
Speak for yourself.
I will try to follow it. And if you don't follow it, don't let us continue. Try to understand.
תודה רבה.
The dorsal neural tube, dynamic setting for cell fate and decision.
Cell fate decision.
תודה.
Okay, so let me immediately introduce you into a romantic atmosphere, and I thank you for inviting me to the Safra Center to tell you a little bit about what we are doing, our thoughts and views. Can you hear me, Micha? לא? אז מה אני עושה? אפשר להגביר כזה? I can. לא, אין לי למעלה.
יש פה ווליום, לא?
This is the closest I can, זה בסדר? איכשהו? Okay, I'll try to speak loudly. Good. So, I'm happy twofold. First, because this is a center to which I belong as well. But second of all, because it's, I think, an opportunity to introduce some of you to some of the concepts and a little bit of data that I'm going to present regarding the development of a very interesting structure in development of the nervous system, which is called the Neural Crest. And without any further ado, before entering into more theoretical concepts regarding developmental neurobiology, where we are headed to and what interests us, let me just give you a little bit of a background to those of you who are tabula rasa in this respect, okay? So again, just as an introduction, the entire nervous system in vertebrates develops from one of the germ layers that is called the ectoderm. And the ectoderm that you can see here undergoes in the midline of the embryo, namely, look at your spinal cord and brain, this would be your midline, undergoes a process of specification into a structure that is called the neuroepithelium, meaning that this structure now, which is thicker than the adjacent ectoderm, will be spaced to generate exclusively components of the nervous system and not of other systems of the body. So there is a certain restriction in the developmental potential of this portion of your ectoderm that from this time on we'll know that it is going to generate spinal cord, brain, and also the neural crest, about which we are going to talk a lot today. So in addition to undergoing this process of specification, the neural ectoderm, which is this structure here, undergoes a morphogenetic change. And all that I'm telling you today is the truth, but not all the truth, okay? Because I'm going to simplify things a little bit. So one mode by which the nervous system is formed is by folding of this neuroepithelium, and in fact, the neural folds come together, they approach each other until they close and fuse in the midline to generate something which is very simple, a neural tube. In fact, this is a means by which many tissues in the embryo are formed, and the neural tube, or the nervous system, is not an extraordinary feature here. Your gut, the entire respiratory and digestive system, is formed via a very similar mechanism, namely the formation of a very primitive tube from which many organs are going then, cell types and so on and so forth, are going to develop later on. So what we can see here is a folding neural epithelium. As you see, this is thicker than this region on both sides. This is going to become the ectoderm or the epidermis, the skin that is going to cover the nervous system, and this is going to become the neural tube. Now, I would like from this moment on to point your attention to a structure or to a group of cells which morphologically is completely indistinguishable at this time point from the rest of the epithelium, a group of cells that are sitting here in the interface between the superficial ectoderm fated to become epidermis and the neural ectoderm that is going to become the nervous system, and these are the neural crest cells. Now, once these neural folds come and oppose each other, they fuse in the midline, the ectoderm regenerates. You can still see some scar tissue here. This is a scanning electron micrograph. And you have a new neural tube, which, as I mentioned before, is going to generate the entire spinal cord, neural crest, and your brain. Now, also, everything looks the same because the neural tube is what we call a pseudostratified epithelium. It's never mind, an epithelium with very particular characteristics. What we can say is that molecularly speaking, there is already a segregation, a diversification of some fate within this structure. And the neural crest, which is our issue today, are these particular cells in dark blue, which transcribe a factor, a transcription factor, which is a DNA binding protein, in this case named SNAIL2 or SLUG in previous years. We know of about more than 30 transcription factors and different receptors that are expressed and characterize this particular region of the dorsal neural tube. Now, as you may know, the dorsal neural tube, after the formation, production, and as you will see in a minute, delamination and exit of these cells, which will form the entire peripheral nervous system, the dorsal tube forms the roof plate, which is an organizing center very important to determine different interneuronal types in the nervous system, both brain as well as spinal cord. So the dorsal neural tube has to be seen as a kind of very dynamic structure which changes with time. And this is what I'm going to try and convey to you during this talk. So just without entering obviously into any detail, just notice what are the derivatives of the neural crest. This is a very, very discrete cell population which comprises a very few number of cells at the beginning of its ontogeny, but later on diversifies both qualitatively as well as quantitatively into an enormous amount of cell types. Just to mention some of them which are relevant to us, obviously neurons and glia of the sensory ganglia, the satellite cells within those ganglia, the autonomic nervous system, which comprises all the neurons and glia in sympathetic ganglia, parasympathetic ganglia, the entire innervation of your intestine, and the entire digestive and respiratory system comes from the neural crest. So many, many neurons and glial cells. An additional cell of interest are the Schwann cells lining along peripheral nerves, endocrine and paraendocrine cells, of which adrenomedullary cells, the chromatin cells, which are responsible for the fight or flight response through catecholamines, come from the neural crest. Pigment cells, all the pigment cells of your body, except for the retinal pigment, are a neural crest derivative. Interestingly, not a neural derivative. However, in malformations or diseases of the neural crest, generically called neurocrestopathies, we can very often see the involvement of pigment cells. One of them is Hirschsprung's disease, a neural megacolon. It's the distal intestine which is not innervated because of a lack of migration of neural crest cells into the side. And in people with Hirschsprung's disease, in particular, it is an autosomal dominant disease, in particular of Ashkenazi Jews. What we can see is also patches of pigmentation in the skin. So it is something which involves both neural derivatives as well as pigment cells. Another one would be neurofibromatosis, which are tumors of Schwann cells, schwannomas, in which you can see also a very active involvement of pigment cells. This is just to illustrate, and we'll come back. This is going to be quite a central point in the talk. And then something which is evolutionary important, and I'm just going to mention it in very general terms. We know that the entire nervous system derives from ectoderm. We also know that the entire connective tissue, the bones of your body and your muscles, derive from another layer which is called the mesoderm. However, in the head of all the vertebrates, it appears that all the bones of your head, of your neck, of your maxilla and mandibula are of neural crest origin. Something which is called the mesectoderm, a kind of chimera between mesoderm and ectoderm, which has an evolutionary significance, and we'll come to that in a minute. So the message from this slide is not to overwhelm you with data. The message is that you take a very, very tiny population of embryonic cells, and it diversifies enormously to give rise to many, many derivatives in the embryo in a very stereotypic manner. And the neural crest is an invention of the vertebrate embryo, and therefore it has become, during the last decade, one of the most interesting and appealing systems to study developmental biology or developmental neurobiology, because one of our main interests in this field is to understand how a homogeneous population of cells during early development is able to segregate into so many different derivatives. What are the signals? What is the nature of the interactions between the cells? What is the amount of information that is carried on by the cell, sometimes from maternal information, sometimes from cell intrinsic processes, and what is the amount of information that the cell gets from its environment, and how does it integrate all this information to ensure such an output? So these are the main questions we ask, and one of the model systems that we use in the laboratory is in fact the neural crest. And let me then try to conceptualize a little bit on what I said before we enter into the crest, and saying that also all of you deal with the complexity of consciousness, of physiology, of plasticity in the adult brain. What we do is we deal with actually the generation of complexity. This is why I think that, and as we talked with Mona or Elon before this talk, it's very important that people at ELSC and in general, biologists and people in general concerned with function of the nervous system should get some course at least in developmental neurobiology. I think that it clarifies some of your ideas a little bit, may put them also in place. So in terms of generation of complexity, I would like to obviously address development, but let me just convey you a few things about evolution. As I said, one of the important things that the crest does is it creates mesodermal derivatives such as bone, muscle, which is smooth muscle in particular, and a little bit of adipose tissue in the region of the head. But why is this so? There is a very beautiful theory that is called the new head hypothesis, emitted in the 80s by Ganz and Northcutt, and what they say is the following. During evolution, we witnessed the cephalization or the concentration of neural structures from a first diffuse type of information processing that takes place throughout the body in invertebrates to something that's both physically and in terms of information processing takes now place within our brain. So the brain has grown. Most of the senses have concentrated to the region, to the cephalic and facial regions. And together with this, evolution tells us that predation became one of the main features, namely animals turned from vegetarians into carnivores. They needed very strong jaws to masticate and to digest their prey. So one of the inventions of evolution in the transition between invertebrata and vertebrata that is the uterostomes, is in fact the neural crest. So why is it so? I will not enter into detail, but the theory would claim that with the process of cephalization, the brain, which is a newly emerging huge structure that concentrates so many inputs, needed a kind of protection. Now, the mesoderm, because of reasons I'm not going to detail now, which normally generates bone, is unable to generate bone in the head of the embryo. Completely unable. For those of you aficionados, there is no sonic hedgehog, there is no notochord, there is no induction of PAX1, PAX9 in mesodermal tissue, so there is a complete inability of the formation of bone and cartilage in that region. So evolution says that there is a co-option of different functions, and in this sense, the neural crest has undertaken the role of creating the skull of these embryos. This is a chick, okay, a skull of a chick, and what you see in red is the contribution of the neural crest to the skull of a chick. Okay, so it's enormous. It's a huge amount with very, very few bones coming basically from what we call the paraxial mesoderm. And if we bring this to the human, what we see is that many of the bones of the human being, except for the basiocciput and except for another occipital bone, are all derived from the neural crest, including the mandibula and the maxilla. So this is a very important issue, very difficult to demonstrate, but with very nice evolutionary correlates. So let me just go back, if I may, to develop a little bit the issue of ontogeny and of development. So evolution contributes to the generation of complexity, but also during development, this is exactly what we are dealing with. And when I'm talking about development, I'm basically talking about the generation of cellular diversity. You saw this huge list of derivatives of the neural crest. We talk about the mechanisms underlying the specification of a cell to decide what is going to be its fate during adulthood. And this is a decision which I'm going to show you today is taken much earlier than previously thought. Then, pattern formation. Cells, having decided or not yet decided what they will be doing in life, have somehow to form a pattern. We have bilateral symmetry, we have a liver in one side, we have ganglia segmentally organized throughout the body. These are patterns. The cells do create patterns by migrating stereotypically, and we have to understand how does a cell migrate? How does a cell know when and where to stop migrating? How does a cell know that it has to coalesce within the peripheral nervous system into a ganglion or into a nucleus within the brain, etc., etc. These are the questions we are trying to understand. Finally, these cells are going to differentiate. Namely, they are going to express de facto the entire amount of transcription factors or the entire degree of specification that they have. For example, if we take a muscle cell, it would express MyoD and MIS5. These are master genes for myogenesis. However, the cell is still a myoblast. And it is only when it becomes a cell expressing tropomyosin, desmin, actomyosin, etc., and it becomes a fiber that we can call it a differentiated muscle cell. So there is a huge difference, obviously, between phase specification and differentiation. These are two consecutive and sequential events. And finally, morphogenesis. I think this is trivial. The formation of a form which is distinct in different aggregates of cells, in particular in the nervous system, when every aggregate is composed of different cell types, different neuronal types, and obviously also different satellite cell types or glial cell types. So this is all about, and this is a crash course basically in development, in which developmental biology and neurobiology are obviously coming together here. Developmental neurobiology is only a branch of biology, and the amount of interactions between the tissues is enormous. So you cannot be a good developmental neurobiology if you don't understand development in general. This is one lesson that I learned years ago. This is why we also work with other systems, not only on CREST or CNS neurons. Okay, so once the basic feature of the dorsal neural crest has been established in the neural tube, and you have to look at the tube as a kind of baguette that is pointing to you. So this is a cross section. It's like I'm slicing the baguette into different slices. And you see here the pseudostratified epithelium. These are epithelial cells that you can see also here, attached by cadherins and so on and so forth. And then begins a very dramatic event for the lifetime of a neural crest cell, which is an epithelial-to-mesenchymal transition. Mind you, this is a very, very widespread phenomenon in development in general, and this is one of the highlights that transforms a cell which is very rigid and very connected to its neighbor, that only proliferates and expands, into a cell that is able to engage in movement, in migration. And what do these cells do? They begin migrating, they leave the neural tube, they invade the entire embryo through pathways that are highly stereotypic. So, years ago, I wrote with my former mentor during my post, many years ago, a book, and I thought that I knew everything about the neural crest. And once writing the different chapters, I became acquainted with the fact that nothing was known about the actual process of EMT, epithelial-to-mesenchymal transition. So I decided that we would like to begin understanding this process, and in fact more than 10 years, and Maya Groisman is here, she also joined us at a certain point. Now she joined Givat Ram, and we began trying to understand how are these cells undergoing this epithelial-to-mesenchymal transition. This is again an SEM, a scanning electron micrograph. The epidermis has been removed. You can see the dorsal tube. You can see these little beautiful cubes called somites, which are going to become your vertebra and your muscles. Okay? And what you can see is that neural crest cells, these beautiful cells here, begin leaving, they emigrate, they undergo this EMT, epithelial-to-mesenchymal process. They leave the neural tube and they migrate into the somites that are the substrates for migration and organization of all the ganglia. And what you can see is that this EMT is graded along the axis. Here, as we approach the head, which is up there, there are many cells, but their number becomes fewer and fewer as we go to the still unsegmented region of the embryo. This is approaching the tail, and other things are happening in this area of the embryo. So we had to understand what are the factors that account for such a graded behavior and a graded delamination of cells. Delamination, cells that leave a lamina, cells that become mesenchymal. And in fact, I'm not going again to explain this. This is not the center of the talk. What we have found in almost, I think, 10 years of work is a molecular network of very intricate interactions between different molecules, and the only one I'm going to mention is BMP4 and Noggin. So BMP4, bone morphogenetic protein 4, is a morphogen. It's a molecule which acts at different concentrations, leading the cells to become different phase, pretty much like Sonic Hedgehog, for those of you who heard it. In addition, it has other functions, such as being the master regulator of EMT, of delamination of neural crest cells. Now, it turns out that BMP4 is expressed throughout the entire axis in the dorsal neural tube. However, Noggin, there is a molecule called Noggin. It's an inhibitor of BMP. And these two molecules, the molecule and the anti-molecule, live together in the very same cells. However, they create a gradient. Why? Because BMP basically in transcriptional terms is constant along the axis. However, Noggin is extremely concentrated here, thereby inhibiting activity of BMP there, and no crest cells can come out. However, Noggin disappears as the somites differentiate, as these structures differentiate. BMP is released from inhibition, and a full cascade of events begins taking place here. And these events involve, obviously, breakdown of cadherin intercellular interactions, the breakdown of Rho GTPases, which hold the cells together, Wnt signaling, and a lot of other things that I'm not going to talk about. So what we have done in a first stage is understand the molecular basis leading to this graded exit of cells from the neural primordium. The question that remains open from this study is what was the state, or what is the state of commitment of the cells that delaminate from the dorsal neural tube? Are these cells multipotent? Are they able at the single cell level to generate each and every of the derivatives of this axial level, or are they already fate restricted? Are they already cells knowing what they will become? And what we know about these cells is in the area of the trunk, we work between the hind limb and forelimb, or between the wing and the hind limb of the avian embryos. What we know is that these cells form their sympathetic ganglia, Schwann cells along peripheral nerves, dorsalward ganglia, and then the melanocytes, the pigment cells here. Now, what you see here in green is the result of one of the systems that we use that is called electroporation. It's a system by which we can deliver genes, either overexpress them or delete genes from different structures using a spatial and temporally regulated type of mechanism. Okay, I'll come to that a little bit later. Basically, what you do is you inject into the lumen of the neural tube, into this hole here, you inject DNA. In this case, we have injected DNA that encodes for green fluorescent protein, for a protein that fluoresces in green. So DNA is a negatively charged molecule. And if you put your embryo, okay, or you put electrodes within the egg, okay, it's very easy to work with avian embryos because you take your embryo from the incubator, you open a window, you have direct access to the embryo that is sitting in front of you, and you can put it in an electric chair, namely between a positive and a negative electrode. And you zap it, you apply electric current, and then DNA is driven to the positive electrode. The electroporation creates very little holes within cells that are very tiny microscopic holes into which DNA can enter into the cell. The holes seal immediately, within seconds after electroporation, and what you have is a kind of transient transgenic animal, namely an embryo that will express in its cells a foreign gene, and this is the foreign gene, GFP, okay? So, using this technique, what we asked is whether, the first question, whether there is any order in the colonization of these different derivatives. Now, just look at it. The entire hemineural tube is green, but the only cells that live are the neural crest cells, so we don't care about these ones, which stay within the epithelium, okay, just as a kind of remark. So what we asked is whether there is any kind of order in the colonization by these green cells of the different derivatives, or whether this is done at random. And in fact, what we found is that there is a highly patterned order in the colonization of the cells, meaning that if we electroporate the neural tube at a very early stage, okay, this is somite stage, this is one way in which we can determine the age of the embryo. If we do it at a very early stage, when the entire cohort of neural crest cells is still sitting in the dorsal tube, what you get is green cells in all the derivatives. The electroporation technique is a mosaic technique. Not all the cells are electroporated. You have an efficiency of about 70%. If you do this at a later stage, you begin losing green cells in sympathetic ganglia. If you do it a little bit later, you lost the sympathetics and the Schwann cells, and so on and so forth, in a way that shows that the colonization of the different derivatives is, in fact, a one in which the ventral derivatives are colonized first, and then the dorsalmost derivatives, namely first the sympathetic ganglia, then the Schwann cells, then the RG, and finally the melanocytes. And if you do it at 44 somites, no more neural crest cells can be stained. So no peripheral structures are seen any longer. So this raised the first important question. If there is order, what is the mechanism that accounts for the replenishment of the cells within the dorsal neural tube? What does happen within the dorsal neural tube that accounts for 2 days of consecutive delamination of cells and colonization of derivatives? And in order to answer these questions, Shlomi Kristin, a PhD student in the lab with now a postdoc at the NIH, emitted 2 alternative hypotheses. The first one is that in the dorsal neural tube you have stem cells, or a kind of stem-like mechanism by which a cell will divide into a green cell that will delaminate and a cell which is red that will remain as a kind of stem cell to account again and again for another round of proliferation and delamination. And an alternative possibility is that at this particular time in development, prior to delamination, there is no more stem model, but instead there are few green cells which are the first to leave the dorsal neural primordium, and once this happens, there are red cells that come into the dorsal midline and will leave in turn, and so on and so forth, until the entire cohort of neural crest progenitors has left the neural primordium and the peripheral nervous system is formed. In fact, what I would like very briefly to show you is that this is the mechanism, and that the stem cell mechanism, I'm not going to enter into this, accounts in fact for the first expansion of the very, very primitive crestals, but not for the behavior that we see during the process of emigration of cells. So if this would be correct, then you should expect that if you mark the dorsal midline with CMDI, a lipophilic marker, or with GFP, that one cell leaves, but the other cell remains in the tube. So there is always residual labeling in the tube, if this is the mechanism that holds. And mind you, this is not what happens, because you mark the dorsal tube with a green or a red mark, and after a few hours, all the cells have left. You can see the green cells in dorsal root ganglia, also here, red cells in dorsal root ganglia, and no residual cells over there. You can also mark the cells, and by this I'm trying to give you a feeling of what we do, just as a kind of, again, feeling this area is something like 50 microns. Okay, so we are talking about something which is very, very difficult to tackle experimentally. And you can also look at an embryo from a dorsal side, from the back. You mark different cells in the dorsal midline. A few hours later, everything is gone, cells are here, and no residual progeny over there any longer. So obviously, this source and sink model, as we call it, is likely to be the case, but in order for this to be true, what you have to show is that these red cells, once the green ones have left, these red cells are able to relocate into the dorsal domain, and this is what, in fact, they do. Let me just go over this scheme. I'll skip the actual data. For simplicity purposes, what we do is we mark a ventral area within the dorsal tube, and we can see with time that the cells approach the dorsal tube and begin leaving. So this means that the dorsal neural tube is a dynamic structure. Nobody knew that before. We all thought that the dorsal neural tube, prior to the actual formation of the spinal cord, is a region where cells proliferate, where cells also die, and where cells differentiate. But nobody knew that there is a kind of funnel type of mechanism in which a cell, when it leaves the neural primordium, another cell replaces or takes its place, in turn will leave, and so on and so forth. Try to look at, I don't know, an analogy like a river, in which it looks like a stable structure, but if you look at individual particles, they are streaming all the time. Everything is dynamic in front of your eyes. This is the way I would like you to look at this structure. So progenitors located at ventral sites to the midline progressively relocate dorsalward prior to leaving the neural tube. This ventral to dorsal movement not only involves crest cells, but also roof plate cells. I'm not going to talk about this issue. The main point is that the dorsal tube is a dynamic region sequentially traversed by distinct progenitors. Look at it as a kind of funnel. So if this is true, we should be able molecularly to see the process. What do I mean by molecularly? Let's look at all those molecules or genes that characterize the neural crest. And in fact, we can look at 3 genes, FOXD3, SNAIL2, SOX9, all of them transcription factors, okay, DNA binding proteins. And what we see is that at a very early stage, when all the cells are still in the dorsal tube, the expression pattern of these 3 genes, this is protein, this is RNA, is very broad. This is in situ hybridization. When crest cells begin delaminating, what we see is that the pattern of expression begins to be very narrow already. And when all the neural derivatives have left and only melanocytes are still populating the dorsal tube, those ones which are the last ones to leave, the pigment progenitors, there is no more FOX, SOX, or SNAIL in the dorsal neural tube. So there is a dynamic behavior of the genes that characterize the neural crest pretty much as at the cellular level the way we showed it. So if everything is dynamic, think of it. We propose that there is a fate map within the neural tube. What is a fate map? In developmental biology, a fate map is like the homunculus for those of you who work in the brain. A fate map means that we would be able to predict by the localization of a cell in different areas of the dorsal tube, or by the time it leaves the dorsal tube, we would be able to predict what its fate is going to be. So we proposed that a few years ago, and Shlomi, indeed with Erez Nitzan in the laboratory, demonstrated that this is the case. I'm going to be a little faster and just show you that, for example, if we mark the dorsal neural tube clonally, namely we have developed a technique by which we inject DNA into single cells and are able to look at the progeny of single cells in vivo within the living embryo. If you do this, and this is how it looks 6 hours later when GFP is translated into protein. So if you do this very early, what you find is that cells, the green ones, this is a magnification of this box here, go only to sympathetic ganglia. If you do this 2 somites later, namely 90 minutes twice 2, okay, later you see it in Schwann cells. If you do it a little bit later, it will populate DRGs. If you do it again a little bit later, it is going exactly and only to melanocytes. So there is a temporal fate map. I didn't show you the spatial fate map by which we are able to predict what a cell is going to be via its location in the tube and the time it leaves the neural primordium. So let me just say that what we find is that there is a continuous delamination of cells that is associated with a ventral to dorsal cell relocation. These are these arrows shown here. That there is an inverse localization of crest cells vis-a-vis derivatives. Namely, this cell which is located dorsally is going to become the ventralmost cell, the sympathetic cell. And this cell that is located here is going to become the dorsal cell, a melanocyte. So there is an inverse localization. We also found that prospective roof plate cells, the definitive dorsal portion of the spinal cord, is in fact located very ventrally. And once the cells undergo the dorsal movement, the roof plate closes, crest cells are out, and this is how the dorsal spinal cord is formed. And then there is a spatial and temporal fate map. Finally, what we learned from the discrete derivatives of the clonal analysis is that crest cells are fate restricted. What does it mean? That if we mark a single cell, it will not generate sympathetic as well as sensory neurons. It will only generate either sympathetic or sensory neurons. Now, this does not tell us if the cell is intrinsically restricted or whether restriction is basically dependent on the environment. So this is the question that we are facing now. Still, these 2 mechanisms can account for a fate restriction that is seen in vivo. And in order to try and understand how this happens, and this is basically the model I'm going to try and present to you. We did a series of different experiments that I'm going to describe very briefly. So the idea of cells already fate-specified prior to emigration is a novel idea, because in general, the accepted dogma in the field was that most neural crest cells are multipotent, namely that the cell leaves, it has no information as to fate, it migrates, and then it is instructed by signals of the homing site, for example, a sympathetic ganglion or a sensory ganglion, to become either a neuron or a glial cell, and so on and so forth. And we find that this is not the case. So I think I'm going to skip this. It's a very tricky and difficult experiment. I'm going to go to something which I think is more plastic and show you that one way in which we can basically challenge a cell and ask it, are you multipotent or are you fate-restricted, is by changing its location. This is quite a classical experiment, and let me show you what is the idea. If a cell is multipotent, okay, and I put it ectopically, I take it from place A and put it in place B. If it adopts the fate of the new location, then I have to conclude that this cell is multipotent, that it was naive, and it was instructed in the new location to become whatever. Okay? However, even if ectopically localized, the cell still upregulates a program that is characteristic of some basic or original location, then you would have to say that this cell is at least fate-restricted. And what Erez did is the following. What we know is that the first cells to leave the neural primordium, these are the red cells, migrate ventrally. They migrate from the dorsal portion into the area of the belly, and these cells are all fated to generate neural derivatives, sympathetic, dorsalward, ganglia, Schwann cells. A day later, in the chick embryo, when these cells are already in place, there is the delamination of the last cells, which are melanocytes, namely pigment progenitors. And these are very different, neural progenitors from pigment progenitors. So what Erez did is he took molecularly, and you'll see how, these cells, when they were still in the dorsal neural tube, he provided them with a receptor, which is a guidance receptor, that instead of letting them migrate ventrally, brought them immediately into the dorsolateral pathway and asked the question whether these cells will become melanocytes or upregulate de novo markers characteristic of their original fate. And this is the experiment. Let me just show you some highlights. What we see here, the fixation of the embryos is 24 hours later, before the actual normal melanocytes are appearing there. So what you see is the neural tube. Hemitube has been electroporated, and this is what you see when you electroporate GSP. This is the control. All of the cells go ventrally. No cells go under the skin. Now, if you electroporate EDNRB2, which is the endothelin receptor B2, that is a guidance receptor, what you see is that about 35 to 40% of the cells now migrate underneath the ectoderm. These are marked now by arrowheads. And you ask yourself, are these melanocytes? The answer is no. They do not express MC1, which is a melanosomal marker. They do not express MELM, which is a differentiation marker. Green cells are not red. However, you can see these markers here, a day later appearing in these nice melanocytes migrating there. And they do not express c-KIT, which is again a receptor characteristic of melanocytes. So these green cells here are not blue. So these cells do not appear to upregulate melanocytic markers. So do they upregulate neural markers? The answer is yes. And just look with me here at these ones. These are the controls. Just for the sake of time, this is the population that now migrates dorsolaterally with the DNRB, and you can see that among these cells there is expression, this is the blow-up, of KESH1. KESH1 is the MASH1 homologue. It's very important for autonomic differentiation. ILET1, which is characteristic of sensory neurons, as you can see here, also for motor neurons, but in the PNS for sensory neurons. FOXD3, one of the characteristic markers of the crest, is not downregulated there. TRAC-C, which is a neurotrophin receptor, again expressed there, and neurofilament, which is the ultimate differentiation factor for neurons. So what these cells did is, in spite of being in a completely foreign environment, they have upregulated a program which is characteristic of their original phase that was neural and not melanocytic. And this is to mean that these cells are phase restricted and not naive. Now, you can argue that if we bring them prematurely into the dorsolateral pathway, the superficial pathway, maybe this pathway is not inducive or conducive to melanogenesis yet, because it's a little bit too early. So we challenge the system again, and what I'm showing you is again the same experiment with the same result, but in a much more fancy way. So what we did is we performed quail-to-chick transplantation of a neural primordium of a very young embryo that contained all the crest cells, and took this neural tube that we enzymatically dissected from a quail host, from a quail donor, and put this in a hole that was left after removal of the chick neural tube. Now, the chick host is a very old one. It is a host in which all the neural derivatives are already in place, and what is going on there is melanogenesis. So we cannot use, again, this excuse. We are now challenging the neural cells with a melanogenic environment, and just for you to wake up, this is what comes out if you let these little chimeras hatch. This is a chick control. This is a quail control. This is the Japanese quail, and these are the nice chimeras. Just to remind you that pigment, okay, comes from the neural crest, and this is what has been grafted here. So this is the experiment. The experiment goes like that. We isolated a pink somite stage donor, a very young one, and this is the host. It has 40 somites, a very much older, 1 day older. And this is the way we removed here under the microscope, obviously. Everything is microsurgery using avian embryos. And this is the hole after removal of the neural tube. And then you come and you put your tube. Look how tiny the tube is vis-a-vis or when compared with that of the host. Within a few hours, the tube closes, everything heals, because the embryo is extremely, extremely plastic and highly regenerative. And you ask the question of what happens to these cells. Do they become melanocytes? Do they become neural? The same question with basically the same answer. So the first part is look at, this is what you do again. You section here the baguette. You perform a cross-section through the embryo, and what you see is here. This is the neural tube. How do I know the graft has been nicely incorporated? Because I can stain with a marker called QCPN that will recognize every cell of the quail. So everything that is red comes from the donor. And what you see here is that this red neural tube has produced red cells, neural crest cells, that instead of going dorsolaterally into the melanocytic environment, look at it, they have formed a mini ganglion that coalesced with the normal dorsal root ganglion of the host. And within this ganglion, some of its cells have even incorporated very nicely and upregulated, as you can see here, islet 1. So some of them have even differentiated into neurons. So what we can do here is, again, combine electroporation of a gene together with transplantation. These are not easy experiments, but Shlomi and Erez did these experiments. What they basically did is they electroporated the tube in the quail donor with EDNRB. Namely, they forced the cells. Instead of going to the DRG, they told them, now you have to go to the melanocytic environment, which is the dermis, the subectodermal domain. And then tell me what are you going to become. And let me just show you this. This is the EDNRB. So some of the cells now are going into the dorsal domain, not only to the ganglia. And what you can see is that in spite of going now to the dermis, to the lateral domain, they will still express and maintain FOXD3. Look at these cells. These green cells are also going to be neurofilament positive, Schwann positive, this is what you see here, and islet 1 positive. Okay, so again, in spite of being totally diverted into a foreign environment, the cells know what they are. So crest progenitors keep their neural identity, even if forced prematurely into the lateral pathway, or if exposed to a non-melanocytic environment. Meaning that the early dorsal tube at flank levels is a dynamic region, traversed sequentially by phase-restricted and maybe even also specified progenitors. And this is news in the field because of the dogma of multipotency. No multipotent cells at this particular stage. And furthermore, early phase restrictions are cell-intrinsic, and this is very important, and do not depend on the nature of the migratory pathway, because even if we put them in a different migratory pathway, they will still behave according to their intrinsic program and not to the program dictated by the environment. What are the mechanisms? Let me try and skip this in favor of the last few slides that I would like to show you. In the last few slides, we'll address the role played by fox, and snail in the pre-migratory neural crest. What is the idea? The idea is basically that if there is phase restriction and there is no dependency, at least initially, upon factors from the environment, then the weight of the phase decisions should be taken back to the dorsal neural tube. So what is special about the dorsal domain of the tube that imparts phase restriction to the cells? We implied and hypothesized the existence of a molecular code, a molecular network, a combination, a crosstalk between molecules that will say if molecules A, B, and C operate, then you will become a melanocyte. If molecules C, D, and E operate, you will become a sensory neuron. This is the idea. We are at the beginning of the road. We are trying to first of all, know all the very many factors that are present in the dorsal neural tube. And what I would like to show you today is what these three factors, fox, and snail, are doing there. And I'll do it quite quickly. The message I'm going to convey you is that these three factors act in the dorsal neural tube as a switch between the production of a neuron or a Schwann cell, mainly generically a neural derivative, and a melanocytic cell. Both of them neural crest derivatives. And I'll go very simply through this. The idea is, as you saw before, that fox, and snail are present in the dorsal tube as long as neural progenitors are produced. However, when neural progenitors left, fox, and snail disappear from the tube, and melanocytes are negative. In fact, what we demonstrated, and I skipped, is that melanocytes initially belonged to the fox lineage. However, they downregulated it during their journey from the ventral to the dorsal domain of the neural tube. Okay, so there is a lot of dynamics within this area. So if fox, and snail disappear in melanoblasts, this means that maybe their downregulation that normally occurs is necessary to enable a switch from becoming neural to becoming melanocytic. So we have to inhibit these three factors in order to be able to switch. And in order to test this hypothesis, what we did, we prevented this normal downregulation. We prevented it in the cheek, and what you get is a disappearance of melanocytes and a reprogramming of the cells into glia. This is the message. I'll go through it very briefly. What you can see is that here in the normal conditions, you get melanocytes. Okay, however, if you apply fox, or snail, this is at later stages, you only hit melanocytes, you do not hit neural progenitors. You see that the cells that are here are MC1 negative, they are not melanocytes, whereas here they are all red, no melanocytes, the green cells are not red here, and they also downregulate EDNRB. You see that these cells here are EDNRB positive, however these ones, the green ones, are EDNRB negative, and negative. So they downregulate traits which are characteristic of the melanocytic pathway. However, they upregulate traits that are neural. They are unable to generate neurons because melanocytes apparently have lost the capacity to become a neuron. However, they are still plastic to become Schwann cells. And in fact, they are able to upregulate P0, and this is what you see here. This is the normal pathway, the normal melanocytic pathway, P0 negative. But look at this pathway with FOX. You can see, I hope, red cells here, red cells here, and red cells there. So continuous expression of FOX, snail, and SOX re-specified melanocytes into glia. Namely, you have to have a normal downregulation of these genes in order to have the normal switch between a neuron or a Schwann cell into a glial cell. And very interestingly, these three factors, which do the same thing to neural crest progenitors, are obviously hierarchically related. We find, and I'm not going to enter the details, that snail acts upstream of SOX and FOX is downstream of both of these factors. So there is a little minimal network of three transcription factors that are able to generate the switch between becoming neural or becoming melanocytic. And this is now the last part of the talk, in which you obviously have to do the opposite. If you overexpress FOX, and clearly what is important is that if FOX is downstream of these other two, this gives us the legitimation to work on FOX, okay? Because all the other two converge into FOX. So, what you do when you overexpress is you eliminate melanocytes and remain with neurons. So the idea or the question is, what happens if you eliminate FOXD3 in the system, namely the converse experiment? In the chick embryo, this is possible. However, it is much more tricky. So what we did is, together with Patricia Labosky from Vanderbilt, we got mouse embryos in which FOXD3, this gene, was very specifically deleted only in the neural crest lineage, not in the entire embryo, because FOX is a very, very important molecule. It is important in gastrulation, so the embryos die very early. So what we did is what is called a conditional knockout, only in the neural crest cells. And Wnt1, as I showed you before, is a marker that is highly expressed and specifically expressed in neural crest progenitors. So it was done under the regulation of the Wnt1. Never mind. I'll go directly to the data. And in fact, what we see, the prediction is that if overexpression is melanocyte to neural, loss of function will lead to the entire converse phenotype, namely an upregulation and overproduction of melanocytes at the expense of neurons and glia. And this is what we get. So the first thing is, when we look at early development, you see the dorsal tube. This is the mutant. This is an heterozygote. What we see is MITF. MITF is the microphthalmia gene. It's a master regulator of melanogenesis. In order to become a melanocyte, give the cell MITF, and even a neuron is going to become a melanocyte. So this is a very important gene. And TUJ1 is a neural marker, never mind. What you see in the mouse embryo is the appearance of the first melanocytes at a late stage, but look at the mutant. The mutant upregulated MITF already in the dorsal neural tube. We never, ever can see this in a normal embryo. Namely, the dorsal neural tube becomes melanogenic already, very early and prematurely. Now, what we also see is that crestals undergoing migration become MITF positive. And in the ganglia, you can see massive melanogenesis. This is seen not only by MITF, but with a battery of melanocytic markers that I'm not going to show you. So instead of having neurons in this DRG, you have melanocytes. You never, ever can see a melanocyte in a normal ganglion, okay? This is seen also at the cranial levels, namely in cranial sensory ganglia, the jugular, the petrosal, the trigeminal. You can see beautiful melanogenesis at the expense of neurons and glia. So it has completely reverted the programming of the cell. In addition to that, you can also see that in the peripheral nerves, Schwann cells have become melanocytes. So this is a control. This is a cranial nerve 11, the accessory nerve. You can see a beautiful staining of neural markers, no melanocytes. However, in the mutant, look at this. Many, many blue cells, many, many melanocytes spread on the nerve, actually on the nerve. This is very reminiscent of a kind of neurofibromatosis type of phenotype. And in the peripheral nerve at spinal cord level, exactly the same. You can see no melanocytes in a normal spinal nerve, a lot of melanogenesis in the nerves. So FOX is important. FOX and snail are important for fate restriction of neural crest cells, and this is my summary slide that presents an interim model of fate determination in the pre-migratory domain of the neural tube. So what we claim is that we can basically subdivide the spinal cord progenitors into two stages, an early neural stage and a late melanocytic stage. In the early neural stage, there is a minimal network at least, and we are working now on expanding and understanding the full network of snail SOX and FOXD3, FOX acting downstream of the other two transcription factors, that has a dual type of information vis-a-vis different progenitors. It promotes a neural identity, however, it inhibits, this is the sign for inhibition, it inhibits the advent of MITF, the master regulator of melanogenesis, and of EDNRB2, which controls lateral migration. By the way, these are separable processes. I'm not going to enter into this. At a later stage, once the neural progenitors have left the dorsal primordium, there is a factor that inhibits the expression of snail SOX and FOXD3, a factor whose identity we still ignore. And when this happens, the inhibition on MITF and EDNRB is released. MITF induces melanocyte identity. EDNRB promotes lateral migration. And we have demonstrated that it's enough to misexpress in any type of cell these two genes to get a melanocyte. So there is a switch between neurogenesis and melanogenesis in the neural crest taking place before migration in an environment-independent manner, and this is given at least by a minimal code of transcription factors. So this is what I wanted to tell you. And last but certainly not least, the people who perform this work are mainly Shlomi Crispin and Erez Nitzan. Erez is still in the lab trying to cope now with the molecular network underlying fate restrictions. And let me mention also Maya, who is here in the audience and moved to Givat Ram. Maya was a postdoc in the lab. She contributed a lot to our understanding of the process of EMT of neural crest cells and the involvement of Rho GTPases, which are actin cytoskeleton, modifying molecules, Tirit, Chauvalon, and Kadhirin, Yehia Kassem, who worked a little bit with Shlomi. And as I said, you cannot be a good neurobiologist, at least not a good developmental neurobiologist, without understanding other parts of the embryo. And several people in the laboratory are working on the ontogeny of the somites, the development of the musculoskeletal system, which is not less amazing. And parts of the work that I did not describe to you were performed together with Avihu in our department and with Trish Labowski for the mouse study. Thank you.
So the jaw bones and the smooth muscles come from lateral migration?
Basically, they come from the migration of what we call mesencephalic neural crest and diencephalon, posterior diencephalon and entire mesencephalon. So the migrations are, let's say this is your mesencephalon, the migrations are both transverse as well as rostral, and they go and they cover your entire brain in order to generate
including the jaw.
Well, to produce the jaw, this is not the mesencephalic crest, this is the rhombencephalic crest, coming from the hindbrain, from rhombomeres, from hogs expressing rhombomeres, 4 to 7.
But still from the ectoderm.
Absolutely. Absolutely.
And in the intestines, the same.
And what? What is the same?
Intestines are lined with those lateral migrations or not?
Well, the intestine is extremely interesting. What happens in the intestine, as I said, is that you have all the enteric innervation, which is subdivided into the myenteric plexus and the submucosal plexus, Meissner and Auerbach plexi, is entirely formed by neural crest cells in addition to sympathetic and parasympathetic innervations. So what happens there, assume you have the intestine, which is a tube at the beginning, okay? Very, very simple one. So this is the neck, what we call somites 1 to 7, something like in this area. And then you have all the somites here, and you have sacral somites, let's say 28, 35. So what we know is that the innervation of the inner plexi of the gut arrive exclusively from somites 1 to 7. Now from 7 to 28 nothing happens, and from the sacral area, namely somites 28 to a little bit more caudally. So what the cells do at the area of so all your innervation comes from your neck. This is the message, okay? Okay, the innervation of your stomach, your intestines, your liver, everything comes from your neck. So what these cells do is that they, this is the neural tube, okay, they leave the dorsal neural tube, they migrate until they reach the gut. Again, this is the prusa in the sandwich. This is the gut. Once they arrive into the gut, the cells associate with the mesenchyme of the gut and migrate. And they migrate up to the tail. And then, while migrating, they proliferate like crazies and they leave behind neural ganglia. Okay? And then the sacral crest does the same. However, the sacral crest does not colonize the entire length of the gut. It colonizes it until the area of the umbilicus. So what we have is, the area of the midgut. Okay? So what you have is a double type of innervation of the hindgut and a single vagal innervation of the entire enteric nervous system. But it's very funny and very special, an absolutely fascinating thing.
You've shown that cells are essentially committed for a phase before they migrate, right? So this would imply that their migration trajectory needs to be very accurate, because they're already committed, they need to get to that place. So do you know how this is done and how this is...
I didn't speak about that, but the intuitive approach would be, and some people claim this is the case, that if you know who you are, okay, you also know where to migrate, through and to. It appears this is not the case, okay? And this is what I tried to allude when I said that melanocyte identity and lateral migration can be separated. This can be basically, taken into molecules. Now, melanocyte identity is MITF, etc., etc., etc. Lateral migration is ED and RB2, at least in the chick embryo. Okay, in other embryos, these are other molecules like c-KIT and ED and RB. So this would imply that if you upregulate MITF, you automatically upregulate ED and RB. This is not the case. It appears that the regulation of MITF is cell intrinsic, and the regulation of ED and RB happens only when the cell emerges from the neural tube, and ED and RB is under the regulation of endothelin, of the ligand of ED and RB that is provided by the environment. So once the cell leaves, it finds the ligand, it upregulates the receptor, and dynamically it begins responding to this ligand by enhancing receptor expression and then crawling through the pathway. So this is what we believe happens. In any event, we can upregulate in a neural cell MITF and transform it into a melanocyte. However, it will migrate into a dorsal root ganglion. We can upregulate, and you saw it, EDNRB, bring the cell into the lateral pathway, and it will still be a neuron. So these two events, counterintuitively, are obviously a cell that is a melanocyte will upregulate EDNRB, but they are separable experimentally. So they are regulated differentially, which is not intuitively. So, Masha.
The last experiment, which is so dramatic, where you forced neurons in the dorsal ganglion to become melanocytes. I forget what was done by snail too, or done by IMRIC.
Fox, by losing Fox.
And then you forced them to become melanocytes, and the evidence for that was they were expressing melanocyte markers.
Right.
If I remember right, you didn't show that they stopped expressing neural markers. And the question is it possible that they still express receptors for NGF and for neurotropins, and maybe the melanocytes are growing axons?
Are meshuge? No. No. I didn't show the data. These are still cooking. This is a work that we are preparing for submission, and the answer is no. These melanocytic, basically, in early development, these are quite exclusive traits. The moment you begin upregulating MITF, you inhibit FOX. Okay, so there is a cross inhibition between MITF and neural markers. So we have tested for brain 3, we have tested for TRAC-C, we have tested for for different glial markers in the mouth, BFAPP, and others, and they all disappeared. Now, what happens to these dorsal root ganglia? Believe me, they don't survive. Okay, a melanocyte with time cannot survive within a neural structure. Okay, so this the fate of these cells within a few days, but this is what happens, is to die.
Tumor cells at least do very well in the brain.
Well, yes, but what you see in normal development of this mutant is that there is a 50 to 70% decrease in the total amount of cells, and these are the melanocytes which undergo apoptosis, but they do so later on, not at 10.5, 11.5. They do so from E12 and onwards.
So you can't be a melanocyte and a neuron.
Eventually not, but you have touched upon a fantastic idea. Plastically, in culture, you can take a Schwann cell and it can become a melanocyte normally. You can take a melanocyte and in culture make it to become a Schwann cell. So the plasticity between at least Schwann cells and melanocytes exists, and we know that it comes from a single cell. But this is plasticity under conditions in which you remove the cell from the normal environment and from the constraints to which this cell is restricted to within the embryo. It happens in a dish when you give the cell with a lot of growth factors. So it reverts. However, you cannot revert a melanocyte into a neuron. The neuron, once, and you saw it, when we apply FOX or SOX or snail, the melanocytes, the late ones, become Schwann cells, become P0 positive cells. However, they never, or at least we, never saw them differentiating into a neuron. We can reprogram them partially. This means that a melanocyte in the dorsal tube has irreversibly lost the neurogenic ability. However, it has kept the gliogenic ability. And there are many examples for glia, melanocyte things, and half of it I didn't present to you. There is a beautiful experimental paradigm in the peripheral nerve that I would be glad to show you, still unpublished data. Mona.
So the question now would be, what in the natural environment prevents multipotency?
Okay. This is obviously the key question, and the idea that I have is that there are cell-cell interactions of an inhibitory nature in which a cell, and well, I'm not going to show you in the interest of time, maybe, obviously, nobody knows what happens, but there are very, very close interactions between one cell and the other, and once a cell leaves, the nature of the interactions of the others begins changing. What we think happens there is that all these changing interactions between cells are generated by a gradient of a morphogen. and I have to do it. Just 1 second, it will be much clearer like this. I skipped this slide, which is obviously very important. The question is whether specification, which is obviously the other side of the coin to multipotency, happens via a spatial mechanism or via a temporal mechanism. Namely, you could argue that the cells have a color coding, meaning specification already in the dorsal neural tube, or you can argue that the cells are still multipotent in the dorsal neural tube, and only by the time they approach the dorsal midline, they become different. We have excluded this. We have excluded this by conditional inhibition of dispersion of cells, in which when you regain disinhibition, the cells still go according to their original pathway. So we don't think this is the case. I can tell you what the experiment is. However, we think that there is already specification here. Why? Why do we eliminate multipotency? We think that there is, and this is hypothetical, a gradient of BMP4. And different cells along the dorsoventral axis here become exposed to different concentrations of BMP4. For example, the yellow cell is exposed to high BMP4, whereas the red and blue cells will be exposed to a lesser or to a lower concentration of BMP4. And BMP4 is instructive. So if you have an instructive mechanism within the dorsal tube, pretty much as you have sonic hedgehog in the ventral tube, that determines in an instructive way the development of ventral cells into floor plate and distinct type of motoneurons, then what you end up having here is loss of multipotency and the beginning of differential specification to distinct lineages. So this is hypothetical, and this is basically the model that we are trying now to demonstrate. And I call upon people who know and like to do modeling. This is a very nice project to do some theoretical modeling of how many progenitors do we have, how do gradients in development indeed end up in making differential phase specification. So this is the idea. So this is why we think cells are not multipotent.
All right. The last question in the interest of time.
These cell determination, are they restricted to the neural tube, or does it happen even before in the neural plate?
This is a very good question. We don't know. The question is, how early is fate segregation, or at least fate restriction, taking place? Basically, you have to argue that at a certain point in development, still in the neural plate, when the first crest cells begin appearing, there should be at least, this is our view, but again, it's theoretically, they have to create a critical mass of cells that begins then specifying.
Simple, when do they start expressing the snail and the fox?
Well, but snail, fox, and sox only discriminate between neural and melanocytic. They do not discriminate between subneural progenitors, such as sensory and autonomic traits and stuff like that. But it is theoretically a simple experiment. We haven't done that.
But what about the neural fold?
We haven't done that. It's the same question. The question is, how early does it happen? What we did is we worked on the closed neural tube prior to delamination, just because people said that long after delamination and during migration, cells are still multipotent. And it's much easier working here, because don't forget, every cell is 10 microns. We have to impale single cells. Imagine that you go to a neural plate. I mean, it's technically extremely challenging. This is very challenging. This is a project that took more than 5 years just to begin impaling single cells and do horrible statistics of hundreds of embryos. So this is very difficult. So go to the neural plate. Very difficult. But the question, obviously, conceptually is correct. How early can we trace back fate restrictions?
Okay. Thank you very much.